Okay, welcome back. So we're going to wrap up chapter 2 with these slides right here. So we're going to continue talking about some of our key macromolecules, and now we're going to talk about proteins. So proteins have so many roles when it comes to your body. They're going to have a role in metabolism. They're going to have a role in signaling, transport, structure, movement, defense. There's all kinds of different roles that proteins have. It's a key component of your diet because the individual subunits travel throughout the body and they have so many functional roles. So you should appreciate them. They're really great. So let's identify what's the monomer and the polymer and talk a little bit more about them. So the monomer is an amino acid right here. So an example is you're going to see that there's going to be several key groups or key parts to an amino acid. There's going to be what we call an amine group right here. You're going to have a central carbon, the hydrogen off it. You're having a carboxyl group, and then you're going to have this R group. Now, this R group is super important. The reason I say that is this is what's going to give the unique characteristic to each amino acid. So if you look at it, all of this part is the same between each of these. Same. But if you look at these R groups, look how they're unique and different between each of them. That's going to make each amino acid have its own unique qualities. Now, when we want to go through and take one of these monomers and start building a protein, we're going to make what's called a polypeptide. So polypeptide is multiple amino acids. So the monomer is an amino acid. The peptide, or the polymer, is a polypeptide. Now, it gets that term because the type of bond formed between two amino acids called a peptide bond right here. And this is going to happen in the ribosome, and we're going to talk about that when we talk about translation. So as I mentioned, that R group gives unique qualities. So depending on what the protein or where the amino acid is and how the protein is folded, it's going to determine what it's going to do. So this is just a table of the 20 essential amino acids, and you can see a lot of variety. So depending on what the R group is, it can make it a nonpolar molecule, give it a positive charge, a negative charge, make it a polar molecule. If you remember when I talked about the structure of the membrane, so we have the part that loves water and doesn't love water. So the membrane protein that's going to go and touch both the water-loving and water-fearing parts is going to have a combination of polar and nonpolar molecules. Okay. Now, when we talk about protein structure, it's all going to talk about how we take this long chain of individual amino acids that we made into a polypeptide and how we're going to fold and bend it on each other. You use the term structure equals function. So remember when I talked about the carbs and how the long chains were organized to determine what kind of function it was going to do. So if it was very linear and straight, it gave a lot of structural support versus if it was kind of loosey-goosey, it was just for quick energy. Now there's going to be 4 levels of protein structure that you're going to talk through with this in this book. So the first is going to be the primary. The primary is just a sequence of amino acids. Really straightforward. Very simple. It's just starting from this point all the way to the end. That's a primary structure. Now the next level up is what we call the secondary. So the secondary is when we start to take this chain and we're going to start to kind of twirl and fold it in on itself. And what happens is those hydrogens that are sticking out and those R groups, side groups, are going to start to interact with each other. And when it does that, it forms 2 different combinations, either called a beta-pleated sheet or an alpha helices. You can have a combination of both within one single protein. So we're going to have this first level of folding over and over, and it's all going to be thanks to interactions between those side groups and the hydrogen bonds. Now, the next one is the main structure we think about proteins is this tertiary structure. It's the 3D shape of the polypeptide chain. So we'll start to form these secondary structures, and it's going to keep folding in on itself till it gets to its final 3D conformation. Now, the way to get to this is there's going to be interaction between those R groups and those hydrogen bonds. So they're going to keep folding and interacting with each other until we get to this overall 3D shape. a lot of proteins stop at this point right here. As I mentioned, you can have a combination of both alpha helices and beta pleated sheets in your tertiary structure. And then finally, some reach the level called quaternary. As I mentioned, not all proteins go to this level. This is a combination of various tertiary structures coming together. Examples are hemoglobin or collagen. So we have individual protein subunits that are coming together to make the quaternary. So primary is just a chain. Secondary, we start to interact that chain with those side groups as hydrogens. And tertiary is a 3D overall shape. So we keep folding and folding, and this is going to be its final shape. And if you have several subunits that come together to form a function, it'll reach quaternary. Now with protein, form equals function. So if it's not in the right shape, it probably can't do its job. So if we alter the shape, we do what's called denaturation. So this is what happens if we take a protein, so here it's final arrangement right here, and we unravel it. So think of it as like this is like a big knot, you know, to make its final shape. Denaturation is like unraveling a protein back to its primary state. Now it's lost its function, now it can't do its job. So it's pretty much a useless protein. Now there's several things that can cause this. Salt, adding high heat, changing the pH concentration can cause this to go from this nice 3D shape to its primary structure. Now sometimes this is permanent. You can't do that. If you ever think about an egg, so you know eggs are high in protein. When you add heat to an egg, you can never go back. It's never going to go back. Some proteins, if you remove the heat source or salt, put it back in the right environment, it can fold right back up. Specific on each protein. All right, finally, the nucleic acids, the last big macromolecule. So there's two types that we'll kind of talk about, RNA and DNA. So RNA is going to have multiple roles in the cell depending on the type of RNA it is. So you're going to see mRNA, tRNA, rRNA. And when we talk about transcription translation, these will make a little bit more sense. And each of these are going to have a different structure, and that's going to lead to the overall function. Now, RNA is super important because if you don't have RNA, you can't make proteins. So it's one of those key things. You got to have RNA in order to pretty much make anything that you need to survive. And you're going to see that we make RNA based on DNA, which is kind of cool. Now, DNA is going to be your instruction manual. It's going to have all the recipes that make everything in your body. So regardless if you're single-celled to a complex organism, you have to have DNA. And pretty much it's a storage place, you know, it's a cookbook for you. Now, these two do play off each other. So you have to have DNA in order to make RNA, but RNA to make some of the components that DNA needs. So very important, these two are very cool. So let's talk about the monomer first before we talk about the polymer. So the monomer for the nucleic acids is called a nucleotide, and it's going to have three main components. You're going to have a phosphate, sugar, and depending if you're DNA or RNA, it's going to be ribose or deoxyribose, and then a nitrogen base. Now this nitrogenous base is going to vary depending if you're DNA or RNA. So this is just kind of showing you some of the structure right here. But this is showing you each individual monomers that we have for the nucleic acid. And when we start to assemble them and to form these chains is where we're going to get to this double helix, which is DNA. So let's talk about how we assemble them. So the polymer is multiple nucleotide units. So what's going to happen is we're going to create what's called a phosphodiester bond between the two individual nucleotides. So let me get my pill. So here's our individual monomer right here. And we're going to start to grow this chain. So what we're going to do is we're going to create a bond that's going to happen right here between this monomer and this monomer. Switch back right here. So this bond between each of them is called the phosphodiester bond. It's very important. What's unique about it is we can only add more nucleotides into what's called a 3' end. So you'll hear DNA is anti-parallel, and I'll show another picture about that in a second. But when it comes to growing DNA, we can only grow it in a certain direction. So we can only add on the 3', so I can only add nucleotides this way. And for this strand, I can only go that way. So when we go through this process, we're going to have two individual strands. So DNA is kind of like a ladder. And what happens is these bases like to pair with each other. And what they're going to do is they're going to form hydrogen bonds with each other. So if you think of DNA kind of like a ladder, boom, 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 the outside part of the ladder is the sugar phosphate backbone. And the inside part are like the nitrogenous bases interacting with each other. Now, depending if your DNA or RNA will determine if you're actually going to be double-stranded or single-stranded. So this is where those base pairing rules. So as I mentioned, the bases like to pair with each other and they have complementary basing, and it all depends on the shape. So they're only going to pair with the ones that match exactly. It's like putting a puzzle together. If you're talking about DNA, adenine's always going to pair with thymine. And if you're talking about RNA, DNA also, guanine's going to pair with cytosine. They're always going to match up together. Now with RNA, what's unique is we do not have thymine. We have uracil in place of it. And genes here are always going to be the same. Now this is going to be super important when we get to Unit 5. And then just remind you their bases are held together by those hydrogen bonds. So this is like a good example of showing you how it's like a ladder. All right. So here is one sugar phosphate backbone on this side, and here's your other one on this side. So as I mentioned, these sugar and phosphates are like the outside part of your ladder on both sides, and the nitrogenous bases are like the rungs, and they interact with each other. Now, as I mentioned, it's also anti-parallel. This just has to do with how DNA is organized and how these nitrogenous bases are able to interact with each other. So we're going to have what's called anti-parallel. So if you look at the ends, we're going to label them 5' and 3'. And I'll go into more detail when we get to the DNA anatomy section in Unit 5, I think. So you'll hear the term anti-parallel. That means the strands are opposite on the ends right here. So this top part of the strand, you'll have a 5' and 3' end, and it'll be opposite for the opposing end. What's going to happen is this is now going to twist on itself, and it can be right or left-handed depending on what the type of DNA is, and it will kind of give its overall structure. So what's great is it's very complex, and like I said, this is the instruction manual for you, but thanks to these complementary base pairing rules, both strands can serve as a template when it comes to DNA replication. Now, RNA is usually going to be single-stranded, and that means it can fold in on itself and form these things called hairpins and loops, and we'll see more about that when we talk about transcription and translation. One of the things it doesn't have that base uracil, it does have the base uracil, but that replaces thymine, and it has a different sugar. And it's that sugar that kind of gives that backbone a lot more flexibility. So this is a good comparison of the structure. So we have the 4 bases for each of them and the overall structure. Now, structure equals function. As I mentioned, RNA is going to have a lot of different functions when it comes to transcription and translation, and DNA is going to be the pretty much instruction manual, but they do play off each other, and they're both super important. Okay. And the last one I wanted, just a little side story, is who discovered DNA. So when you think of DNA, you might not know any names, but Watson and Crick might come to your mind. There's also a fellow named Maurice Wilkins who had a key role in this too. But then there's also a silent figure that really didn't get much credit. So her name is Rosalind Franklin, and she actually took the first X-ray photo of DNA. And when this was happening, it was a very hard time for a woman in science. I think it still is. And she was doing, I think she was doing some work in Wilkins' lab, and he pretty much matched her up with another student, said, here, work together on this. And she came up with this first picture, which is really cool. The same time that she's doing this, Watson and Crick are working on the structure of DNA, but they're like at a roadblock. They don't have the information to fully publish the data they have. Well, Wilkins is friends with these two fools, and he shared her information with them without her knowing it, which is kind of sad. That was the missing piece that these two needed to kind of fill in the gaps for information. So when this was done, it got published in Nature. They eventually got, their Nobel Prize and all that stuff, but she didn't get credit at the time. It was later on they finally recognized her, but she was already passed away, because the work she was doing is very dangerous. So the takeaway story is get credit for your work, no matter what you do. That's your work. Make sure you get credit for it. So here are a few review videos on macromolecules, and that wraps up chapter 2.